I am going to be showing you some very, very new data. We got into, Candace Guerrero at our Center for Mass Spectrometry and Proteomics came to our lab a couple years ago, knowing that we are really interested in metabolomics, and basically showed us this biocrates kit and thought that we could incorporate into our research. So a lot of what we're doing is very new on this front, and we're really excited to expand this. And we think that it's really, I'll show you some really kind of hot new data that really indicate that there is some promise here in what we're doing in the lab. But we've typically done a lot of microbiome and information work. So I'm going to give you kind of a background and give you some of the overview of what we do in the lab. So thankfully, I had a wonderful predecessors in my talks this morning. I was Stuart giving a nice kind of background in microbiome. And so as he alluded to this morning, there's actually, and I think he said the same amount of microbial genes, but I think there's an argument to be made. There's actually more microbes and more microbial genes in the body than there are human cells. I always like to give, when I give talks, the fun dinner party fact that you have 5 to 10 pounds of bacteria in your gut, the normal adult does. So we don't just have these bugs. We don't have all these microbiota in our gut, and the rest of our body. There's several microbiomes which I'll talk about, but we don't have them just, to be there. These are highly active, highly functional organisms that are very much influencing human health. Just to make sure we're on the same page, the microbiome is not just bacteria. I'm going to be focusing on bacteria today. A lot of people do because they're better studied and there's more, a high abundance of them, especially in the colon. But the microbiome includes any microorganisms in a microenvironment. So bacteria, viruses, fungi, protists, archaea. My lab is getting into studying a lot of these other ones, but like I said, we'll focus on the bacteria. But again, these are very functional microorganisms. So it's not just the bugs that are there that's important, but what they're doing. So the genes, metabolites, and products of these microorganisms is part of that microbiome and extremely important in when we consider these. So I always say in the lab, it's not what's there, it's what they're doing and why they're there. An important word is dysbiosis. So this is an imbalanced microbial community, and this can be either abundance or functional changes of our microbiome. And when there's dysbiosis, there's almost always a consequence for human health. So, and this really, I keep meaning I need to really switch these arrows back and forth because there's multiple things that can influence our microbiomes. I think that we, probably know some of the obvious ones like diet, but genetics, environment, lifestyle, hormones. So Carolyn's talk was amazing. Really gave me a lot of ideas of ways we could collaborate, which is why these meetings are always the best. But because hormones are such an important part of this, and this is why there's huge sex differences with microbiomes. And then there's multiple microbiomes, and that are very important across the body and protection from disease, all kinds of things. Like I said, in dysbiosis, when these microbiomes go wrong, it almost always has a cause in human health. So I used to teach a microbiome class back in the day, and I would have this list of diseases that when microbiome dysbiosis occurred that were affected. And finally, I was like, this is stupid. I should have a list of diseases that it hasn't been found to correlate with in some way, and that is basically a blank slide, because virtually every single human disease that has been assessed relative to the microbiome, there's been evidence that microbial dysbiosis can affect human health in some sort. There's many more microbiomes in this. Virtually your whole body has a microbiome at different sites. There's a couple of protected sites, such as the placenta, for example, that doesn't have a microbiome typically, but even in times of inflammation or disease, then can have bacteria infiltrate. And there becomes this sort of inflammatory microbiome. But virtually your entire body is going to have some sort of microbial capacity at some different time in life. A couple of them that we are really interested in my lab is like vaginal microbiome. We do a lot of work on. I'm not going to talk on this today, but happy to at lunch speak more about that because we do quite a bit of work on women's health with this. But really focus on the gut microbiome today because that is the vast majority of microbes in the body and pertinent to what we're discussing. So I would say this is like the Klatt Lab slide. So we have, and my background, you kind of hear, I have this weird, diverse background of, molecular biology and immunology and microbiology and pharmaceutics. And, I've done a lot of adolescent and young women work. And so I've kind of taken all of these years of experience and collaborations together and really kind of made this model of systemic inflammation, mucosal dysfunction. So mucosal dysfunction actually contributes to more diseases than you'd ever think that aren't mucosal related. Like almost every cancer has a component of mucosal dysfunction. Alzheimer's, for example, you think of as a gut. or a brain disease, but there's more neurotransmitters in the GI tract than there are the rest of the body combined, including the brain. And there's a huge component, mucosal dysfunction, even in things like Alzheimer's. Microbial dysbiosis, obviously, we're going to focus on a lot with my microbiome research today. And then immunity inflammatory responses. And there's this whole vicious cycle. So once one of these is disrupted, the others are affected vastly. And then we have this component of drug metabolism I'm going to talk a little bit about today, too. It's a newer, some newer work that we and others have been doing. So, and then the important part about this is it really is applicable to everything. I always kind of make jokes that we're a schizophrenic lab because we work on so many different disease states. But really, I mean, this model for dysfunction and human health can be, virtually any disease state, whether it's we do a lot of work on women's health, like I said, or surgical outcomes and sepsis or wound site infections, cancer and transplant. We do a lot of transplant research now. I'm going to show you some COVID data. My history is mostly infectious disease, so I'm going to talk a bit about HIV. And then antibiotic and antibiotic resistance. All of these things can be vastly affected. And then we're also really interested in neurodiseases as well, and sort of the gut-brain axis. So, having collaborators like Stuart could be a great outcome of this meeting. So just to remind you, a lot of people kind of say, oh, is HIV even a problem anymore? Don't we have drugs for that? But it absolutely is. So we don't have a cure for HIV. People who are infected with HIV, despite having good antiretroviral drugs, still have chronic inflammation and mucosal dysfunction, and morbidities and mortality are much higher, and the actual overall life expectancy is about 20 years lower if you have HIV infection, even if you are, in the US and having good treatment. And of course, we still have new infections every year, and infection rates are going higher, especially after COVID, and young women are actually the highest rate. So it's still really important to study and to understand. The important thing too is that people are on lifelong drugs. These drugs are not necessarily good, and we don't really understand why. This has been a huge topic of research in the HIV field. These are called the HIV reservoir. So despite you can suppress that viremia, and you can't measure active virus in the blood, and we have, people are on, usually 3 or 4 drugs at a time now. But when you take them off the drugs, that virus comes back every time because there's reservoirs, and we don't really understand why. And so this is something that we're going to talk about a bit today. So like I said, this mucosal dysfunction is really important when we think about pathogenesis and this really can apply to multiple disease states, but in this context, we're going to talk about HIV. So in a healthy individual, can you guys see the pointer? Yeah. In a healthy individual or a healthy gut system, you want a nice intact epithelial barrier that's going to protect from any pathogens and, food products and things that are coming in through the lumen here. You want a healthy microbiome to protect that. And then you have these homeostatic cells and cytokines that also help protect the barrier. This healthy microbiome, again, like I said, the byproducts are extremely important. So we're going to talk a bit about things like short chain fatty acids that these bacteria make. And this is something that is, measured by metabolomics. But these are the metabolites that are really important for maintaining that healthy barrier and making sure that the barrier is not breaking down. And this slide is quite old now. It's from 2015. So I really should update this with things like short chain fatty acids. In the context of HIV infection, some of the things that we've identified as underlying pathogenesis is you get damage to this barrier. And the lack of short-chain fatty acids might underlie this because you do need those short-chain fatty acids to keep replenishing the barrier. We know there's this altered microbiome and that this is problematic, and I'll show you some data to this extent. And then on top of it, in combination of all this, you have the bacterial products that translocate, causing microbial translocation. And then you have systemic bacteria or bacteria in your systemic periphery, and that can drive inflammation throughout the entire body. And then you also have increased pro-inflammatory cells and cytokines, things like neutrophils that really, they release all kinds of highly inflammatory proteins and can then further the breakdown of that epithelial barrier as well, and then recruit more pro-inflammatory cells and cytokines in. So you have this kind of chronic inflammatory state, and again, this kind of cycle that I showed earlier with the microbiome, mucosal dysfunction, inflammation, immunity. So we really work at in my lab is understanding the mechanisms underlying this mucosal dysfunction. So microbiome dysbiosis certainly occurs in HIV. It's consistently shown, every study is a little different, of course, as they always are, but there are some overall trends that we consistently see. So in HIV infection, we typically see bad bugs increase. Bugs like Prevotella, this is important. Prevotella is highly inflammatory. It has a very long LPS chain, stimulates every immune cell and makes it as highly pro-inflammatory. We never want to see Prevotella or increased rates of Prevotella. It's almost always associated with some sort of bad outcome. So some like colon cancer, multiple cancers are associated with increased Prevotella, diabetes, etc. Proteobacteria, it's important that this is increased in HIV. So proteobacteria, first of all, have several opportunistic bugs, so like staph, nuclei, but also proteobacteria is important because they're very adherent little bugs. They can get through the mucus, they can grab onto that epithelial barrier. Almost every time we measure bugs that have translocated, they're almost always proteobacteria. So having these highly inflammatory, translocatable bugs that can be pathogenic, not good to have increases in these. And in the meantime, we lose bugs like firmicutes. So these include like our lactobacilli, so our probiotics, our good bugs that we want, that keep pHs low, that are very good at maintaining the barrier and overall health. And then we also lose things like Bacteroides, and like I said, short chain fatty acids are extremely important in maintaining gut integrity and barrier health, and those are also lost in HIV infection. I got to focus much on this. There's this whole story on sexual preference with HIV, and people saying, oh, well, it was already screwed up. It's not the virus, but we've debunked this at this point. Sexual preference can certainly drive different microbiomes. Receptive anal intercourse can be associated with a more inflammatory environment. But when you see, children that have these exact same components of dysbiosis in the microbiome, we're pretty sure that this is an HIV related phenomena, not just a sex related phenomena. So in my lone lab, we have some new data out and these patients, I'm going to show you some data from shortly with the metabolome. Oh, but like, we good? Am I being told to shut up because I just started? You can just say stop. I'm bored you guys already. So we looked at the microbiome in HIV infected versus uninfected individuals. Oh, no, these are all HIV infected in this case. This one we broke into non-responders in red. So these are people that we say despite being antiretrovirals, their CD4 CD8 ratio. So remember in HIV, the CD4 T cells are what's lost. And even if you put someone on antiretroviral therapy very early, they still have lost the CD4. So we use CD4 CD8 ratio as a very good indicator of immunological responders. And so people who have good responses and they maintain that CD4 CD8 ratio, we call responders, and then non-responders are in red. And what you can see is there's a big difference in the microbiome, even within HIV infected people, that's associated with that immune response. And then this is actually, we take some of these bugs out that we find particularly dysbiotic in this case, and then we look at it versus just the CD4 count, the CD8 count, or the CD4 to CD8 ratio. And there's really huge correlations between specific bugs that we think really, might drive this. Again, it's correlation, not causation. We're working on some more validation and mechanistic work on this, but very interesting, demonstrating that, somehow this microbiome is playing a role in immunity. So again, just, going back, just continuing to think about this cycle I showed you. But then on the other hand, these antiretrovirals, they're important for health, but then they can also actually, oops. Sorry, how do I go back? Previous. Oh, I'm just missing a figure here, but maybe I don't know where that is. It's on my other one. Okay. Well, there's nice growth curves here, but you can kind of see in this. But so basically what we do is when we can measure different bacteria that we know are dysbiotic or probiotic and associated with disease or not disease in HIV, and then we measure them with different antiretrovirals to see if there's an impact of the antiretrovirals on the actual bacteria growth. And like I said, I'm missing the growth curve, but really what this is the heat map of that growth curve. So, but we have a series of different bacteria, and different drugs can differentially affect the growth of these bacteria. But then conversely, and we've been doing very close work with the mass spec core. So before it used to be like you measure one drug at a time with this, and now we're developing assays. We're up to 17 drugs at a time, where we actually can do these, and then we can look at the actual, we can quantify the drug level as well, because it's not just important that the drugs can affect the bugs, but the bugs can affect the drugs. So we do these different cultures. So here we have an example of several different bacteria. And then the A-beta control, and what we can see is that different antiretrovirals in culture with bacteria, the bacteria actually can metabolize them. And we showed this with vaginal bacteria several years ago, that vaginal bacteria can actually metabolize drugs used for prevention of HIV, and that can affect the actual efficacy. And so now we're looking at how this might play a role in the context of HIV pathogenesis. And in fact, we show that in culture, we see that certain bugs can metabolize certain drugs. So this is potentially a mechanism leading to this chronic inflammation and potentially HIV reservoirs, because despite having these good antiretrovirals, the actual bugs that are dysbiotic in HIV are actually metabolizing that drug and making it less efficacious. Sure enough, when we look at stool, so now we took stool bacteria, or just a stool, we have stool from HIV infected or unaffected people, and then expose them to these different antiretrovirals. So we have TDF here, dronabinol, or TAF. And what you see is the blue is HIV infected stool, or HIV unaffected stool, or stool from HIV unaffected people. The black is just abiotic controls. And then the red is stool from HIV infected people. And especially with TDF and dronabinol, so two HIV drugs, the people who have HIV infection, actually, the bacteria that are in their stool are highly metabolizing these drugs. Whereas if you do not have HIV infection and don't have these dysbiotic bugs, you don't see as much metabolism, especially compared to abiotic control. And then there's certain drugs like TAF, a newer drug that's a newer tenofovir version, and both the HIV infected and unaffected stools can actually metabolize it. So it's something that's not considered much. And I think Carolyn already had a head of the airport, but one of her colleagues, Andy Goodman at Yale, has been doing some beautiful work where they actually showed that of 300 drugs they measured of any, so they have, SSRIs, antipsychotics, antibiotics themselves can be metabolized by the bugs, actually, antiretroviral drugs, other antivirals, they had, obesity drugs, diabetes drugs, all kinds of things. And they showed of 300 drugs they measured, 270 of those drugs can be highly metabolized by the bacteria from the gut, from stool bacteria. So, it's an important thing to consider. But then this is kind of our model of how we're looking at this and what we're doing here coming up. So we have all these prospective and retrospective cohorts. So some of these data I showed you here. And then we can look at, we've got ileum and colon, and actually we're getting rectum now too, stool, lymph nodes, and then we have these co-cultures in blood. And then we can actually kind of assess like how much these dysbiotic gut bacteria might metabolize antiretrovirals or produce inflammation, so leading to this. And so we have this whole model, but obviously this microbiome-proteome, and again, Stuart, that was really nice that you went before me talking about, how there's like a human versus microbiome. I'm going to show you some from both. But, the understanding this metabolome is a good way to kind of understand potentially some of the functionality and the mechanisms underlying how these bugs can have these different effects. So again, I probably don't need to tell you guys what biocrates is. But we've really been using it, again, for this kind of hypothesis generation when we were doing this, and really validation. And then we can get, specific metabolites and then go track those down and see how, what bugs make them, how this affects things. So here was just really a very, kind of quick and dirty, I think it was 5 HIV infected, 5 uninfected. We could not believe how many things actually came up significantly different. So this was from plasma in HIV infected versus uninfected. So this is really going to be like more host metabolites. This isn't going to be so much of the bugs. But what we see is that a lot of the things that are affected are really indicative of, so there's LPS issues, and then, and this is not lipid polysaccharide, by the way, but there's a lot of lipidomics that are really affected. So we're doing a lot of works with lipidomics now too, and understanding the lipid profiles in HIV, which we know can be changed. And then there's a lot of mitochondrial dysfunction proteins. I just want to highlight a few things here. So PLA2 activity is increased in HIV positive. So this is highly associated with inflammation. No one's shown this before in the context of HIV that we know of, but this has been very well studied in obesity and diabetes, and has been highly associated with inflammation and lipid dysfunction. So this was something that's interesting in just these 5 patients in each cohort. And this is a known phenomena that the lipid profiles have changed in HIV infection. And so, this is starting to get at maybe some of these mechanisms using the, something like the metabolomics to say, okay, like why is PLA2 going up and what's causing this? So that's, again, a path we can go down. On the other side, we have the spermidine. which came up. So spermidine was isolated in sperm, hence the name, but it's actually very anti-inflammatory, and it's actually associated with generation and mitochondrial function and anti-aging even. In fact, it's been, people have been trying to isolate it for anti-aging properties. And you can see it's decreased in HIV infection in the green. These are terrible colors. I'm sorry. I don't know why it's Christmas colors here, but so it's, and these are all significant. Again, this is brand new. Like I literally, like, she wanted the slides like Thursday morning or Wednesday or something. And I'm like, I need these. So sorry that they're not the most perfect slides with the p-values. These are significant. But it's, it was very last minute trying to get some fun new data to show you guys. But really it just, demonstrates that even in these tiny cohorts, it can give us some ideas of pathways to go down. And then you can just see kind of overall when we look at these. So this is going to show you where, your spermidine here. But when we do this PCA-LA, so this is basically, we just take all the data and throw it out and say, is it differentially distributed on something? And it could be anything. We put all the components, we put age, we put everything. And what does it really come out significantly different as? HIV positive versus HIV negative. So it's very clear that these metabolome differences is driven by the HIV infection status, is what this is showing us. And that slide probably should have been first. But again, this is the first time I've ever presented these. So apologies if there's anything. So this was something that we're so excited about. And I don't know why he's got like these different graphs. Again, super last minute trying to get you guys some fun new data. So don't also like go waving about this, because we still have to validate all this and go down the pipeline. But just thought it'd be fun to have, especially since the people who've so generously provided biocrates kits for us to do these preliminary experiments are here. I wanted to show them and highlight the coolness of it. But this was so exciting for us because we're really interested in gut brain, and we have these partners at University of Miami, 2 clinical psychologists that are doing amazing work in anxiety and depression and stress response, in HIV infected and other disease. And again, really interested in things like Parkinson's and Alzheimer's, but we were just shocked and excited to see that when we look at the stool now, this is a stool, not in the plasma. Everything else I had showed you was plasma. This is just the stool, but we see decreased neurotransmitters. So the dopamine is decreased in HIV infection, and then the sum of all the neurotransmitters is decreased in HIV infection compared to unaffected people. So, and we know there's increased depression, anxiety, and stress, but like finding this actually in the gut, like I said earlier, there's more neurotransmitters in the gut than the rest of the body combined. So, I mean, this could have a huge influence on regulation of anxiety and stress and different things, peripherally as well. So we're really excited about tracking this down and understanding this more. So, and then, sorry, one more piece of data too from the gut. So, and this is expected, but always really nice to see validation of. So 3-IPA is, so tryptophan metabolism is a very well-known pathway that the microbiome can influence. So in virtually every disease that the microbiome has been studied, someone has looked at the tryptophan metabolism pathways, and the tryptophan kynurenine pathways are a very clear pathway that cause systemic inflammation coming from gut dysfunction and microbiome dysfunction. And so it was really nice to see that 3-IPA, which is extremely important byproduct of tryptophan metabolism, and when it's when the tryptophan metabolism pathway is disturbed, this is one of the first ones to go down. And so we can validate too, okay, like this mechanism we know can cause inflammation is also there in the gut. So there's also some nice, internal validation things that we've been already described, and now we can validate using this metabolic pathway or metabolomics techniques. So there's clear, this is again very preliminary, but definitely some metabolic dysregulation, both in the plasma and the stool. That neurotransmitter piece is really interesting. So we're really excited. We're actually going to start looking at that. And we have a huge depression trial called Trident. It's a huge program project looking at depression and HIV. So we're going to go and I'm going to, beg, borrow, and plead to Marissa after this about getting kits to, get more preliminary data on this, but see if we can, kind of link some of this neurotransmitter data, for example, with depression levels and things that we know that we're measuring, via psychological techniques. And so, really being able to bind these different fields together is really exciting. How much time do I have left? Do I have a few minutes to go into COVID? 5 minutes? 3, 2, 1. Okay, I'll talk fast. So hopefully I've convinced you kind of, how these different pathways are really important and the cycle is really important. So just really quick, going to show you some COVID-19 data. I mean, we're all sick of COVID. I want to write this up and get it out and never think of COVID again. But just showing though how like the utility of these different pathways and mechanisms, is really overlapping. And of course, really, so we ran the biobank I moved to the University of Minnesota, April of 2020. Just in time for a fun shutdown, right? So my lab actually, when we went in, the first thing we did is forget everything we've been working on, we're excited about, and started this COVID biobank. So we have a really beautiful cohort of individuals we have healthy controls, people who had mild COVID and people who had severe COVID. We have a severity score that we use, and we have multiple different swabs, rectal, nasal, and oral, and then blood and stool, so that we can use for all kinds of different analyses. And we've got a huge storage of this. And then we also have two other sites, Italy and LA, that we have a bunch of samples for. So if anybody does need COVID samples, I am really going to try not to do more work on it. So this is a summary kind of when we took the samples in COVID. So from mostly from June to November in 2020 was the major bulk of when we were taking these. So most of these are going to be in alpha strains. And then we have all of these, right? So they're discharged acute, discharged to rehab, discharged to home, discharged to we don't know, or they died. And then this was interesting too, because we actually included these little tag marks in there as the remdesivir doses, because almost every single one of these patients got remdesivir. So if you remember, it doesn't actually work, but everybody was pretty much getting it back then. Okay, so maybe the Stuart's shaking his head, like maybe there's. I guess it depends on who you ask, but so what was really cool, so we take all these and we did a whole bunch of inflammatory cytokine data, and then we did a lot of gut barrier markers. Obviously, we love the gut in my lab. And we looked at zonulin, which is a measurement of the turnover of the epithelia, LBP, which and soluble CD14 are both two kind of markers of microbial translocation. But importantly, these pro-inflammatory cytokines are associated with length of hospitalization, gut dysfunction, all these different things. And I see you guys checking your watches up there. So I have like a running monologue that I'm going to share with you guys at points. So importantly, rectal swab microbiome alpha diversity was very different in inpatient versus healthy. So if somebody had severe COVID, they actually had less diversity in their rectal microbiome. Diversity is typically a good thing. But, and then we know too that there's specific opportunistic pathogens in COVID-19 that outgrew, and that are known opportunistic pathogens that were growing out in people. And we could find this in oral, nasal, and rectal swabs. So we can find these opportunistic pathogens that were growing out in people with COVID-19. So there's definitely something with the microbiome going on. And then what we did was metabolomics on these, and this is what I just want to show. And so, like I said, so propionic acid is a short chain fatty acid, like I said, is very important for the gut. And and you can measure this in the periphery, and this is definitely decreased in people with COVID compared to healthy controls. And then decanoic acid is another one that we're measuring, and that was actually increased in the deceased, but decreased in people who had COVID and survived. So again, and we don't really know what this means yet. These are correlations, or just kind of brand new data, but again, starting to give us potential pathways to look down to understand, what's underlying all these inflammatory effects. Because still with COVID, we don't entirely understand why there's so much inflammation in some people versus others. So this might give us some insights. And then butyric acid is one of the most probably famous short chain fatty acids. It's very well known, and it's a very, very homeostatic anti-inflammatory short chain fatty acid that's very, very important for gut health. And what we can see, if you look over here, for example, is like IL-12 is negatively correlated with butyric acid. So again, maybe these gut bacteria being dysbiotic and then losing things like our short chain fatty acids is driving that peripheral inflammation. But pathways that we are now currently testing using some really cool new co-culture systems. But just bringing everything back full circle again. It's whatever system we're kind of looking at, there's all these mechanisms, and obviously the metabolome has a very key role in these different things. So we're really trying to expand more into this, and always looking for new collaborations for that. So with that, just want to thank everyone in my lab, Chris Bastian in particular ran a lot of this. He's just an all-star. The Center for Mass Spectrometry and Proteomics, Candice Guerrero, which I know a lot of people in this room know, she's amazing, and she says hi to everybody, and Tim and Bruce, and then I really should have Kevin Murray and Todd Markowski, now they should be on here, that they've really started working with us a lot on these things. So happy to take questions.